Hello everyone, welcome to the new session of Biotechnology in Agriculture. So this is the fourth part of this particular topic. Okay, so we are having some of the advantages of the biotechnology in agriculture. Okay, we have covered some part of it, some part has been left. So let us cover that. Flavr's swar tomato. This is one of the genetically modified tomato. So the commercial sales of the genetically modified food began in 1994 when a collagen first commercialized its Flavr swar late ripening tomato. What is the significance of this type of tomato? GM tomato, genetically modified tomato, that is the Flavr swar variety, that it ripening is late. And this is one of the advantages. Molecular genetic technology was used to slow down the process of softening and rotting caused by the fungal infection. So generally what happened? Tomato is easily get infected with the fungal, right? With the fungus. So it causes rotting and softening of the peel of tomato, right? So what does this particular variety do? The molecular genetic technology, right? It slowed down the process of softening and rotting caused by the fungal infection. Okay, so that lead to the increased shelf life of the GM tomato. It increases the shelf life of the GM tomato, and this is the advantage of it. Additional genetic modification improve the flavor of this tomato. So not only the shelf life of this particular variety of tomato is increased, but also the flavor of this tomato also get increased, right? So the Flavr swar tomato did not successfully stay in the market because of the problems of maintaining and shipping. Next is the golden rice. So the agricultural biotechnology offers various nutritional benefits. We all know that there are so many benefits of agriculture biotechnology, right? So this is one of the advantage of the agriculture biotechnology, that a new variety of rice that is called as golden rice. Now you can see the color of this rice is golden. That is why it's got its name, golden rice. One example of this is golden rice, which contain beta carotene, a vital source of vitamin A for the body. So this contain beta carotene, that is the vital source of vitamin A, means this particular rice provides you vitamin A. Okay, so this is one of the importance of this type of rice. The name golden rice comes from the color of the transgenic grain made from 3 genes, out of which 2 are from the daffodils and 1 from the bacterium. So we are having 3 different genes, right? 2 from the daffodil, this is a type of flower, and 1 from the bacterium. So these 3 genes were cloned to obtain golden rice. Genetically engineered herbicide-tolerant HT crop. So herbicide tolerance, a gene isolated from the gram-positive soil bacterium, that is Agrobacterium tumefaciens. This is a soil bacteria, okay, and it is used genetically engineered herbicide tolerant crop. So this bacteria is very, very important. It has a great use in biotechnology. So it makes the recipient plant tolerant to the broad spectrum herbicide glyphosate. So this is one of the herbicide that is called as glyphosate. So what happened? This herbicide kills the herbs which are present in the crop field. Now along with the herbs, the crop also dies. They also get eliminated, right? So what we do, or what this particular technology did, it has taken some of the genes and incorporated into the plant. and that becomes the herbicide tolerant. So when this herbicide is sprayed in the crop field, it causes harm to the herbs, but our crops are protected. So let us see. HT crops can reduce the cost of production and help in managing weeds. So in this way, we can manage our weeds and reduce the cost of production also. HT crop has been developed under the name Roundup Ready. We have already covered this particular part in one of our session. Genetically engineered herbicide tolerant crops, one more, RR soya beans, were released for the commercial cultivation in 1996. So this RR soya bean has been released commercially for the cultivation in 1996. The yield of RR soya bean is similar to traditional soya bean but reduces the cultivation cost. Although the conventional breeding for the drought tolerant continues to have an impact, it is time consuming and it is restricted by the accessibility of the suitable genes for the breeding. Okay, so this is for the resistance to drought. Some conventional breeding for the drought tolerance continues to have an impact. So earlier we were totally dependent on the conventional breeding method for the drought tolerance production of the crop later, but it is a time consuming process, right? So we adapted biotechnology. This technology makes the accessible of suitable genes for the breeding. So how does it happen? Some examples of the conventional breeding program for the drought tolerance are the development of the rice. So one of the rice, that is drought tolerance, has been developed. Okay, wheat and Indian mustard varieties tolerant to the salt and alkali soils by the Central Soil Salinity Research Institute, Karnal, has also been developed. Development of maize hybrids with increased drought tolerance. So some of the maize hybrid plant has also been developed that is having drought tolerant, right? An effort to incorporate salt tolerance to wheat from the wild related species, right? So wild related species is having salt tolerant, right? So that has been used for the breeding with the wheat. Okay, and incorporation of the drought tolerance as a selection trait in the generation of the new maize and wheat germplasm by the International Maize and Wheat Improvement Center. So like this, maize hybrids and wheat that can tolerate the salt in the soil has been developed by this technology. Through genetic engineering, on the other hand, production of tolerant crops involves identifying the main genetic determinants underlying stress tolerance in plant and incorporated these genes into the crop. So with the help of biotechnology, okay, the gene which is responsible for the tolerant of crops, tolerant for the drought or tolerant for the salt stress, right? So that has been identified from the crop and incorporated these genes to the crop in which we want to get tolerant from the drought or salt. Okay, so the drought causes a wide variety of physiological responses in the plants and influences the function of a large number of genes. Work on gene expression has identified several hundred genes that are either triggered or repressed during the drought. So when they have worked upon this gene expression that is related to the drought, the genes which are related to the drought, now this particular gene has triggered a number of other genes and also repressed some of the genes. Okay, so all these genes has been studied properly and according to that it has been incorporated into the crops. So the gene responsible for the drought resistant has been identified, isolated and transferred to the crop plants using the technique of gene transformation. Mostly two approaches, target and the shotgun approach, aid genetic engineering to obtain the transgenic plants conferring drought resistance. So two approaches has been adapted, that is the targeted and the shotgun approach for the incorporation of the desired gene into the transgenic or the crop plant and making it the transgenic. Okay, ABA dependent gene regulation of the drought stress. So we are having two ABA dependent gene, that is the DREB1 and DREB2 transcription factors. These are the two transcription factors that are essential for the AB independent drought tolerant pathways. So this is one of the pathways that require two transcription factors, that is DREB1 and DREB2, right? So that induces the expression of stress response gene. So in the absence of these two particular transcription factor, right, the expression of the stress response gene will not be possible. So these two transcription factor, that is a type of protein, right? So they are very, very important for the ABA independent drought tolerant pathways. Overexpression of the native form of DREB1 and the constitutively active form of DREB2 enhances the resistance of the transgenic, that is the Arabidopsis plant, to drought, high salinity and cold. Okay, so if there is an overexpression of this native form of DREB1 and the DREB2, that enhances the resistance of the transgenic, okay, Arabidopsis plant to drought, high salinity and to cold. While these genes were initially discovered in the Arabidopsis plant, their presence and role in the stress tolerance have been documented in many other important crops. Okay, such as rice, tomato, barley, canola, maize, soybean, rye, wheat, and maize, suggesting that this is a conserved universal stress defense mechanism in the plant. So if this particular genes that is responsible for the stress, for the drought, for the salt, for the cold from the Arabidopsis, if it has been incorporated in these crops, then our crops can also be AB independent tolerant, right? That is that drought stress. So the BT, now the resistance to the insects. Okay, the BT gene was isolated from the common soil bacterium, that is Bacillus thuringiensis. This we have already covered in one of these sessions, right? So Bacillus thuringiensis and transfer to the cotton, not only the cotton, brinjal also. We also know that there are two, BT cotton and BT brinjal. BT brinjal, right? So BT brinjal has been commercialized in country named Bangladesh, and BT cotton in our country, right? So BT gene was isolated from the common soil bacterium Bacillus thuringiensis and it is transferred to the cotton, right? Which result in the production of a specific type of protein in the cotton that is called as cry protein. Now this protein is lethal to the certain insects such as pink bollworm, that is Pectinophora and Gossypilea, right? And the cotton bollworm, that is Helicoverp azea, and it is partially efficient in controlling tobacco budworm also, that is Heliothrus versens, and the fall armyworm, that is Spodoptera frugiperda. So these are some of the insects that get controlled by the incorporation of the BT gene that is responsible for the cry protein, synthesis of the cry protein. So this particular protein, when get inside these insects while eating the plant, right, they die. And in this way we can control these insects. Using gene transfer technique, the Bt gene responsible for the Cry1 protein from the Bacillus thuringiensis has been transferred to the several crop plants such as rice, cotton, tomato, potato, etc. and insect resistance plant have been developed. So this is all about this session. We will have some more interesting session. Thank you very much.